All right, today we're going to talk about Chapter 11, Antibiotics and Resistance to Antibiotics. Antimicrobial drugs interfere with the growth of microbes within a host. Antibiotics target specific bacteria and selective toxicity are killing harmful microbes without damaging the host. Spectrum of activity. So broad spectrum antibiotics affect a large number of gram-positive or gram-negative bacteria. A narrow spectrum antibiotic is effective against a small range of organisms. For example, penicillin G affects gram-positive bacteria, but very few gram-negatives. Use of broad-spectrum drugs also kills normal microbiota. Survivors may become opportunistic pathogens, and secondary infections can result. Superinfection. Yeast infections may arise from overgrowth of Candida albicans. Another type of superinfection is a term applied to growth of a target pathogen that has developed resistance to the antibiotic. Antibiotic-resistant strains replace the original sensitive strain, and the infection continues. Major action modes of antimicrobial drugs. Number 1, inhibition of cell wall synthesis. Penicillins, cephalosporins, bacitracin, and vancomycin. These drugs all inhibit cell wall synthesis. 2, inhibition of protein synthesis. Chloramphenicol, erythromycin, tetracyclines, and streptomycin. These drugs inhibit protein synthesis. 3, inhibition of nucleic acid replication and transcription. Quinolones and rifampin. These inhibit nucleic acid replication and transcription. And 4th, injury to plasma membrane. Polymyxin B does this. 5, inhibition of essential metabolite synthesis. Sulfonamides and trimethoprim act in this way. Inhibition of cell wall synthesis. Bacteria have weakened cell walls, and eventually lyse or burst. Penicillins, cephalosporins, and vancomycin all work in this way. Here you can see inhibition of bacterial cell synthesis by penicillin. So in box A, we have rod-shaped bacteria prior to exposure to penicillin. And then in B, same magnification, you can see bacterial cell lysing as penicillin weakens the cell wall. Inhibitors of protein synthesis. These target prokaryotic 70S ribosomes, 30S and 50S. They differ from eukaryotic ribosomes. An example of this would be aminoglycosides. Things like streptomycin. It's broad spectrum and it changes the shape of the 30S subunit. These are subunits of the ribosome. So if you change the shape of the kitchen, then you can't make anything in there, right? Tetracyclines are another broad spectrum antibiotic. They interfere with tRNA attachment. Inhibitors of nucleic acid synthesis. Several drugs block DNA replication or mRNA transcription. Drugs often affect both eukaryotic and prokaryotic cells. Rifamycin inhibits RNA synthesis. Injury to the plasma membrane. Some drugs form channels through the cell membrane and damage its integrity. Polymyxin B does this. It's topical and usually combined with bacitracin and neomycin in an over-the-counter preparation. Antimetabolic agents can be effective when metabolic processes of the pathogen and host differ. A good example of this are sulfonamides or sulfa drugs. They inhibit folic acid synthesis, inhibiting the synthesis of essential metabolites. So we've got sulfonamide here on the left and PABA over here. They have very similar structures. Same rings with an amino group, but up here is where they differ. Clinical considerations in prescribing antimicrobial drugs. Safety and side effects. Toxicity. The cause of many adverse reactions are poorly understood. Drugs may be toxic to kidney, liver, or nerves, and considerations needed when prescribing drugs to pregnant women. Allergies are also something to be taken into consideration. Allergic reactions are rare, but may be life-threatening if they occur. All right, so here's some examples of side effects. In box A, we have black hairy tongue caused by antiprotozoan drug metronidazole or flagyl. It's temporary and harmless. And B, discoloration and damage to tooth enamel caused by tetracycline. Resistance to antimicrobial drugs is another very important concern. The development of resistance in populations. Some pathogens are naturally resistant, and resistance by bacteria is acquired in two ways, right? We learned about this in the last chapter. New mutations of chromosomal genes or acquisition of plasmids by transformation, conjugation, and transduction for antimicrobial resistance. Antibiotic resistance often results from misuse of antibiotics and selects for resistant mutants. Misuse can include using outdated or weakened antibiotics, using antibiotics for a viral infection like the common cold or other inappropriate conditions, right? Antibiotics are effective against living things, but viruses are non-living, they're abiotic. So antibiotics shouldn't be effective anyway. Using antibiotics in animal feed. It's often common practice that animal feed is laced with antibiotics, not to keep sick animals healthy, but because a side effect of certain antibiotics are improved growth rates. Failing to complete a prescribed regimen. So a patient is sick, they go to the doctor and are prescribed an antibiotic, but they feel better halfway through the course, so they stop taking the drug. Using someone else's leftover prescription and improper disposal of antibiotics. These are all misuse that can lead to a selection for resistant bacteria. There are at least 6 mechanisms for microbial resistance to antibiotics. Some resistant bacteria produce an enzyme that destroys or deactivates the drug, like strains of methicillin-resistant Staphylococcus aureus. Some bacteria are able to slow or prevent the entry of drug into the cell, like altered porins, and they can change their structure or an electrical charge. Some bacteria are able to alter the target of the drug so it binds less effectively. This is often seen with antimetabolics. Some bacteria are able to alter their metabolic chemistry. They abandon the metabolic step that's being targeted, or abandon the synthesis of folic acid and absorb it from the environment. Antibiotic resistance can also come from pumping the antimicrobial drug out of the cell before it can act. Pumps can often pump out more than one type of antibiotic. And some multi-drug resistant bacteria are capable of pumping 10 different drugs or more. Some resistant bacteria also form biofilms to prevent drug diffusion. So antibiotics are only effective against bacterial treatment. Influenza, flu, and common cold are viral infections, and therefore antibiotics are ineffective. They should never be prescribed for these conditions. How are multi-drug resistant bacteria treated? What if through multiple attempts of treatment there are no antibiotics that work? Sometimes we can find synergistic effects. So if we use multiple antibiotics at the same time, that can overwhelm even resistant bacteria. Might lead to amputation to physically remove the infected part of the body from the rest of the body, or development of new drugs. However, there are no governmental agencies anywhere in the world that are still working on developing new antibiotics. It's not profitable for private drug companies to do it. So at the moment, there are no new antibiotics being developed. All right, effects of combinations of drugs. Synergism occurs when the effect of two drugs together is greater than the effect of either alone. So here's an example of synergism between two different antibiotics. So this is a disc with the antibiotic amoxicillin, clavulanic acid. And this is a disc with the antibiotic aztreonam. So we see cloudy bacterial growth streaked all over the plate. There's a zone of inhibition around each of the discs, but there's also an area of synergistic inhibition, a cleared area between the two discs that's even larger in diameter than the zones of inhibition around any of the single drug discs. True or false? You can become immune to antibiotics if you use them too much. Good. False. You don't become immune to the antibiotic, but your bacterial community within your body can become resistant to it. How about this one? True or false? Methicillin is resistant to methicillin-resistant Staphylococcus aureus, or MRSA. Good. Another false. It's not the drug that's resistant. It's the bacteria that can resist the drug. Be careful you avoid these common pitfalls. All right. So you should be familiar with antibiotics, how they can be used effectively, what we need to do to ensure that they continue to be effective and discourage the development of resistant bacteria. Okay? All right. Come prepared with questions for lab or office hours.